Okay, let's go ahead and pick up where we left off. So we were talking a little bit about chapter 1. So we kind of just got done with all these big biological questions of life. We talked a little bit about how everything is moving toward the ultimate goal of a living organism, which is reproduction. And so everything kind of pulls in that direction. And so what we're going to be doing now is we're going to basically go through this list. So this is kind of like our list for the next few slides as we kind of work through this and unpack these a little bit. So the first step we're going to be going to is the organization of cells. So basically living things are organized, so they have to be organized, right? In order for you to be able to do cellular business, you have to have organization about yourself, right? Because ultimately there's only one requirement for a living organism in life, and that's to reproduce. So how you get there and how you do it and what you do to do it is completely up to you. There's any number of different versions of how to do things to get to that point of reproduction. That's the only requirement in a living organism, is to propagate the species, basically to reproduce. It's one of the reasons why you have so much biological diversity out there. There's so many different organisms, because each organism that does things a little bit differently basically turns into a different kind of a species. And so that's kind of what gives us our diversity. Now, when we take a look at the organization of life, we're taking a look at this a little bit more specifically here in just a second. But we have levels of biological organization. So we have ways to organize this. And the very, very bottom of all of this, of all living things, is basically organizing our building blocks. So what we're actually made out of. So all things in the universe, whether living or non-living, whether on the Earth or in some far-flung corner of the galaxy that we haven't, or the universe we haven't even approached discovering yet, they're all made out of one thing. They're all made out of atoms. Everything, all matter, physical substance, things that have physical substance as opposed to energy, all are made out of atoms. And so that's true for all of us. And so typically speaking, our atoms are organized in nature into pure substances, which is essentially collections of these pure atoms that we refer to as elements. And so those elements are actually up here on the periodic table behind my back. So those are all the naturally occurring elements, plus a few others that we manufactured in labs. But those are the elements that we see in nature and in the universe itself. But we all basically are made out of atoms. Now, typically what happens is atoms will associate with each other, and they will form combinations with each other to basically create molecules. So for instance, if you take two atoms of hydrogen and they form an association with oxygen, what we end up getting here is the molecule water. A fairly simple molecule, but a very, very powerful and very important molecule as well. And so here you have two atoms of hydrogen and oxygen coming together to form a completely different thing, right? And so that's true of all atoms, and that's true of all molecules. And typically speaking, we have different sizes of molecules. Water is a fairly small one. And then we can build bigger and bigger and bigger molecules. We'll talk about this as well a little bit later in actually the next chapter, which is chapter 2. And so we can build things like the biological molecules of life, right? So things like DNA and proteins and things like that actually go into building molecules. Those are called macro molecules because they're really, really big assemblages of atoms that have very specific goals and roles. And so ultimately then you also organize atoms. Ultimately, you don't get there immediately, but ultimately you'll get to the organization of the cell. The cell is the important thing, because essentially what we refer to the cell as the basic unit of life. Now, what does that mean? It means nothing until I describe it, right? So what we mean by the basic unit of life is that this is the smallest that we can get and still answer all the big biological questions of life. Remember that list we looked at last time? Energy, organization, homeostasis, growth, development, reproduction, heredity, all of those are the big biological questions of life. You have to answer those, right? You can't just opt out. Every organism has to have an answer for those. And so the cell is the smallest unit that we can get to and still be able to answer all those big biological questions. Now, some cells are single cell, like prokaryotes, which are like bacteria, single cell, things like that. Others, like us, are multicellular. That basically means we're composed of many, many cells. And if you're composed of many cells, guess what you have to do? You have to organize those cells. You have to get those cells to work together and to kind of create some order associated with them, right? Nothing comes from chaos. Nothing good, that is, comes from chaos. Okay. So that cellular organization is something that really is required in order to be functional, because ultimately the first task of reproduction is to live long enough to reproduce, right? So survival is a big chunk of biological existence. The next level up from that is the tissue level. So basically a tissue is a group of cells fastened together, riveted, if you will, together, and they basically have a particular unified function that they do, okay? And so that's kind of their next level. So you're organizing your cells into tissues, and then ultimately you'll move your tissues into organs. So this is kind of what it looks like. Here's your kind of your pictorial, if you will. So we start off, where's your atoms go? There we go. So we start off with our atoms. So everything basically in the universe is made up of atoms. Those will come together in association to form molecules. And then those molecules will get bigger and bigger and bigger to form the big biological molecules that we think of, those big macromolecules. And those will eventually form the structure and the basis of the cell itself. Yeah. Organelle is actually below cell. So there's actually a step between molecule and cell where the organelles fall. Technically speaking, in a thorough list, it would go from molecules to macromolecules to organelles. And then the organelles will assemble to form the cell itself. Okay, so the reason why they left organelles out is because they want to leave room for those cells that don't have organelles, the prokaryotes. But like, for instance, this is an organelle, right? That's a chloroplast. So there are these, the little green guy right there. So this is a plant cell with chloroplast in it. And an organelle is simply a membrane-bound structure inside typically a eukaryotic cell, which is like us, right? And they focus on one of those big biological questions of life, not all of them, right? So a good example might be mitochondria. Mitochondria is an organelle that focuses on the energy question of life. But by itself, it doesn't answer all the questions of life, just one of them. That's the reason why a cell has to have an assemblage of all these organelles to answer all the questions. That's why the reason why it's the cutoff point, the small, so we can get and answer all those big biological questions of life. So cells then can be grouped and fastened together to form sheets of tissues. For instance, you can see here neurons that are interconnected with each other. I like this one better because you can see the actual plant cells that are actually glued together to kind of form these sheets of cells, and that gives you a tissue structure. And so that's basically what we're made out of. From tissues, we move up to the next level, which is the organ system. So an organ is actually composed of several different types of tissues, usually. Okay. Now, I want to go up to organ system, and I want to use an example of liver to illustrate what we mean between the tissues, the organs, and the organ systems. So when we get up to the organ systems, an organ system is a group of organs that work together on a common purpose. You can see there's kind of a repetitive definition going on here, right? Because essentially you're doing the same thing. You're a collection of something that has a common purpose, but what happened is you're leveling up, right? Okay, so now when we take a look at organ systems, by the way, just to kind of let you know, and I always like to do this for every single group. Typically speaking, if you're in a classroom and your focus is on atoms and molecules, chances are you're in a chem class. Okay. If you're focusing on things like cells and the events and the mechanisms of the cells, then you're probably in some sort of a cell bio class, kind of like ours. Well, sort of like ours. We do cell bio, and then we cover a lot of other things as well. But like a cell bio class is kind of where a general bio would start, right? So we basically start with that cell bio speak. And if you park it there, you're kind of in a cell bio class. We're kind of more of a survey class. But when you put cells together and you're talking about tissues and organs and organ systems, you're probably in an anatomy and physiology class. So like if you're pre-health, pre-nursing, that's kind of where you would end up going. If you're in an organism class, if you're focusing on the organism, it's what's called organismal biology. Chances are you're in kind of a bio 2 class, where you're focused on just individual organisms and how the organisms are structured and work. If you're looking here at species, population, community, and so forth, you're probably either in an ecology class or like an environmental class. So that kind of gives you a breakdown of sort of how this sort of level of organization sort of stratifies each of the different biology classes that you see in any given biological department. So what I want to do is I want to give you an example of an organ system and the tissues. And I want to use the example of the liver. right? So the liver is a complex organ. It's got one function. Its job is to metabolize the nutrients that you absorb after you eat them. But it itself is composed of multiple tissues. So in the liver, you'll have the liver tissue itself, right? So the hepatocytes, the actual liver cells, the ones who are doing all of the metabolism that you associate with like metabolizing whatever it is that you eat. In addition to that, however, there's also vascular tissue in there as well, because you got blood vessels and things of that nature, and that's a different type of tissue. You also have connective tissue in there, because the liver has multiple lobes, and you got to kind of keep them all together, and you use connective tissue sheets, kind of like contact paper, to sort of glue those lobes together and to kind of keep them all in one place. So you have now an organ with at least 3 separate types of tissue that goes into that organ itself. So that's a perfect example of an organ. Most of your organs are not made of one type of tissue. It's made of multiple types of tissues. Okay. Now the liver itself has a role and a goal in the digestive system. So that's the system that it's part of. But it is a member of the system, amongst which are other members, things like the pancreas is also part of the digestive system, and also the endocrine system, but the digestive system really a lot. The stomach, intestines, the rectum, the esophagus. So those are part of the digestive system. And the goal of the digestive system is to digest and absorb nutrients, right? And then of course the liver metabolizes those nutrients before it releases it to the body. So that's an example of how an organ made of multiple tissues resides in a system that has multiple organs. That makes sense? And so that's kind of it gets a little crazy, a little weird in there. And then when you basically collect a bunch of organ systems like us, you end up getting an organism like us, right? So we are collecting basically nervous system, endocrine system, digestive system, cardiovascular system, respiratory system, reproductive system, the immune system. So we have a bunch of systems that come together, and that forms us, the individual. Okay. Now, once you kind of move beyond organism, then you get to basically the idea of species. Now, species and population are very similar. It's just a matter of how you look at it. So basically, a species is essentially a group of interbreeding organisms. Right? Like for instance, us, we're Homo sapiens. So we are a group of interbreeding organisms. Homo sapiens can breed with other Homo sapiens. Okay, so we're a species. A population, typically, is when you have a group, typically a large group of the same species. So for instance, we represent a population of 1006 students. Okay, so that's our population. We're kind of all the same. But there's other populations in the room. You're like, there is? Kind of creeps you out, right? It's like, where? We'll talk about that. Because the next level up, community, is actually taking a look at populations as they interact with each other. So we have one population in this room, Homo sapiens. That's us. We know that, right? But we also have a population of arachnids, spiders. They're like, where? Well, they're here because they're everywhere, right? Or we have a population of ants, or we have a population of cockroaches, right? So if we have all of those, then there's multiple populations in the room. If that was the case, we'd probably be calling somebody, right? So we have multiple populations, and we're interacting with each other, right? So when populations are interacting with each other, oftentimes this is what we focus on, like when you watch those nature programs. This is where we start to see things like predation and feeding, right? So when a cow is eating grass or hay, that is one population, the cow, interacting with another population, the grass. Then when the predator comes in and kills the cow and eats it, that's another population interacting with the population in carnivory, right? So all of these feeding things that we see on nature programs, the lion attacking the zebra and eating the zebra eating the grass and so forth, those are all population interactions. That's a community, okay? Now then, when you add one other thing to that community of interacting populations, so you have a community of populations that are interacting with each other, and then you add to that, not just interactions amongst themselves, that's what's called biotic, bio for life, right? Life interactions, but now you have what's called abiotic or non-living, non-biological interactions. That's with your physical environment, right? For instance, if you start talking about things like soil and say, for instance, rainfall and sun, right? All these sorts of things. Now you're talking about non-living physical parameters of your environment. Once you toss that in there, now you're talking about an ecosystem, right? So basically, when you're talking about an ecosystem, you're looking at, say, for instance, well, this soil doesn't have enough nutrients in it to support our grass for our cow to eat. Now you're talking about an ecosystem. Or like, for instance, if you go up into a mountain range, for those of you hikers who like to get up there, right? If you go above tree level, there's a point at which the trees stop growing. Why? Not because they don't want to, but because when you get up there, most of your soil is not amenable to sustaining roots, right? So ultimately what you're getting at that particular point is rocky soils at best, or just sheets of granite that are impenetrable for most root systems. So you don't basically have any growth above that. When you're talking about that, you're talking about ecological parameters, okay? As opposed to river systems, right? Mountains tend to be very dry because all the moisture is bundled up into ice and it's not available to water those root systems. So that is also an ecological problem. We all know that, by the way, don't we? Because in the wintertime, we all get dry and lizard-like, don't we? Our skin starts cracking. We start bathing in lotion to keep our skin from bleeding too much. My knuckles get terrible. It looks like we've been in a bar fight all winter long because I don't usually use lotion. I just deal with it. Right? So the moisture's locked up in the atmosphere, in the precipitation and ice, and it's not available to you to use. Right? And so conversely, if you take a look at a river system, how many of you guys have looked closely at things like the Cherry Creek corridor or the South Platte corridor? Or if you've ever come from an area where there's big river systems, like I come from Sacramento, that was one of the Bay Area to Sacramento. And so in Sacramento, you have two major river systems. You've got the Sacramento River coming through the middle of the city, and you got the American River coming out of the mountains. So two major river systems. And when you look at it, they're incredibly lush. I mean, you got trees and bushes, willows, and blackberry bushes growing wild everywhere. But there's a lot of life. If you look closely at the Cherry Creek corridor, just right over here, or if you look at the South Platte, pay attention. Where are all the cottonwoods growing? All the cottonwoods, those big gnarly trees that put out all that cotton fluff in the summer that makes us all sneeze and curse for a few months out of the summer. Those are all growing along the waterways, aren't they? They're parking their keisters right next to the Cherry Creek. They're lined all along the South Platte. What that's called, that little area of lushness around river systems, is what's called a riparian habitat. We have a name for that, ecosystem-wise. But that is an ecosystem. So you have populations that are interacting with their non-physical environment, the extra water that's being delivered to them, irrigation style, by this waterway system. It's permeating the soil in and around the river, which is allowing them to sustain the root systems of the willow bushes that are there, or the cottonwoods that are there, or in some cases, to lesser extent, oaks that are in that river corridor. Right? So you can actually see how that non-living parameters is affecting life. And so that's the same thing we have here. So here you can kind of see we're off here in Africa. So we left off at organ. So here you can see your brain versus your leaf. So we had actual leaf cells that were fastened together in tissues, and they form the organ, which is the photosynthetic organ of a plant, which is its leaf. That's a cross section of a leaf. And then, of course, this is just one system associated with the organism. So the nervous system is just one that forms the individual person or the individual tree. Tree is composed of root system and the photosynthetic system, vascular system. There's all sorts of systems in a plant as well, and that forms the organ. And then a species is where you have interbreeding individuals. Notice typically we think small scale when we're thinking species. And then if we think larger scale, large groups, then we tend to think in the population terms. So you can see you've got more of these individual populations. And then the community is the individual populations interacting with each other. So for instance, the Homo sapiens interacting with the trees, interacting with the grasses, interacting with the zebra. And so that's the interacting populations. And then we kick it up and we add ecosystems. So now you're probably thinking, wait a minute, what did we add to the ecosystem? Well, look what we added here. We added water source. That's going to be watering the soil in and around that little pond or that little lake, whatever it is. That's going to allow the zebra to come to their little watering hole and drink the water. That's an interaction of zebra with their non-living environment. That is ecosystem level. For us, we have the mountains up there, right? That's a totally different ecosystem, creating different weather patterns, different moisture patterns, different types of soil patterns, and things like that. That's all ecosystem. Now, when you put a bunch of ecosystems together, you get the biosphere, which is easier understood as basically the Earth. So the Earth is basically a collection of ecosystems. Notice what we didn't talk about with ecosystems. We did not talk about size. Normally, we think of ecosystems as big, because the first ecosystem that a lot of people come to is Amazon rainforest, right? Or something like that. Because we studied that ecosystem a lot, we're still trying to figure it out. There's a lot going on in the Amazon rainforest. Probably some stuff that we're not going to know for generations, really, very well. But one of the things that we tried to do, because we had this concept of size, like we had this question we wanted to answer. And part of the reason why we wanted to answer is because it was an offshoot of conservation. Because back in like the 80s and the 70s and 80s, we were at war with the logging industry. Because at that point, the logging industry hadn't sort of turned into its more responsible version, which is what we have today. They were mostly clear cutters, which means they would just basically annihilate entire old growth forests and just tear them apart. And then they would just leave destruction in their wake. And so conservationists were at war with the loggers. And that's kind of where you get to these stories of the so-called tree huggers, right? Where people would drive stakes into the trees because it would basically ruin the chainsaws of the loggers, or they would chain themselves to trees so that they wouldn't cut them. And so there was a war going on. And the real question was, in conjunction with the Amazon, because they were clear cutting the Amazon like horrendously. And we've lost a lot of tropical rainforest. We continue to lose it. It's a problem. But there's so much you can do with Brazil, right? It's hard to tell another country, stop cutting, and while we're just cutting our forest down. And we've cut the majority of our forest down, actually, in the US in the 19th century. What we wanted to know is we wanted to say, listen, our point was, listen, when you cut down a forest, what you're doing is you're damaging important ecosystems. Species are going extinct, and those extinctions eventually are going to catch up to us. We're going to lose these things forever, and we're never going to get them back. So really, when you take a look at conservation, modern day conservation, which is typically ecologically driven, there was a lot of intertwined concepts that sort of had this main reasoning, this narrative running through it, right? For instance, we have the endangered species list because we had species that were basically facing extinction because of things like clear cutting and stuff like that, habitat loss. And on the other side, we had habitat conservation because we were losing this, we were losing species, and it was basically nothing good was going to happen from this, right? And so there was a war. So the big question was this: how big is an ecosystem? So you want to preserve it. Okay, how much are you talking about, right? So if you want to preserve this old growth forest, like how big is the circle you're going to draw around this forest, which is the no cutting zone? Well, and that's basically spawned an experiment in the 80s called the minimum critical size experiment, which was, what is the minimum size of an ecosystem to be able to maintain a healthy ecosystem? And so there's a lot of work happening there. And unfortunately, the answer that we came back with was not satisfactory, because the answer we came back with was, we don't know. We don't know. Because what we realized was ecosystems can be immense and big, like the Amazon rainforest. But we also realized that an ecosystem can also be a microsystem. It can be as big as the back of a beetle. So what we realized was that ecosystems have macro systems, and there's also micro ecosystems. Like when you're looking at microbial, like bacterial ecology, their life in the soil and things like that, their ecosystem is a micro ecosystem. So basically there is no defined size of an ecosystem. And that's the reason why the general conservation now is basically a general push to say, listen, we need to be responsible with our earth. And ultimately the logging industry changed. So now they don't do clear cutting anymore. They do selective cutting, harvesting, what they call it. But now the forestry or the logging companies actually are very aggressive in reseeding. So they realize that it's not in their best interest economically to just clear cut forest, because at some point there's going to be a day when there's no more trees. And their business is gone. So they realize that there was, as an agricultural entity, that no other agricultural entity just annihilates their crops without replacement. So they have since created a very strong, robust replacement strategy of reseeding, along with selective cutting, so that they can maintain the health of forests. And so they're much, much more responsible these days. Okay, so that is your organization of life. Now let's go ahead and take a look at our energy, right? So basically energy is defined as the capacity to do work. What kind of work? Biological work for us, right? Your cell has to do a lot of work. You can imagine that your task is to reproduce. In order to get there, there's a lot of things you have to do, right? There's a lot of tasks you have to do. Guess what? None of those come for free. So you have to work for each of those. You have to pay for each of those. How do you pay for them? With energy. So you have to lay down some energy currency in order to get some work accomplished. Same thing for us, right? We don't have energy, we're not doing much. We're just laying there like a vegetable, right? So you need energy in order to be able to do your work. So when we are basically looking at this energy system, when we eat, right? So when we eat, a lot of people don't realize this. There's only two reasons why you eat. Biologically speaking, right? Food network notwithstanding. There's really only two reasons why a biological organism eats. The first one is building blocks. You need building materials to build your body. All living systems are made out of the same stuff. So the stuff that we eat is the stuff that we use to build our own body. You literally are what you eat. That's true for everything, okay? The other reason why we eat is to get energy. We need energy in order to be able to build the molecules that build our body. And so we need nutrients. Basically, nutrients are required for the metabolic reactions. In our body. And we also need the building blocks to build our body and the energy to fuel those reactions. Okay? So that's the reason why we eat. We don't eat because it tastes good. We eat because we need materials, we need energy, and we need nutrients to make our systems run. Okay. So our metabolism, which basically is defined as the lump sum of all the chemical reactions that occur in a cell or an organism, that's our metabolism. And so ultimately that kind of is a lot of going on there. So we're not going to get into that. It gets into biochem basically. Ultimately, that's our metabolism that's happening. And our energy that we're getting from our metabolism has one source. And our one source of all energy in all living systems on the earth is simple. It all comes from the sun. So if you think about all the energy that's in all the organisms on the face of the planet, every single one, not just the 9 billion of us, but all the grass, all the trees, all the insects, everything, which by far outnumber us. All of that, if you distilled it all down into one big energetic pot, all that energy comes from one place, the sun. The sun is the one that drives all the energy. It's the one that feeds the energy into the earth, right? Which is one of the reasons why when we're looking for Goldilocks planets, we're looking for planets that have proximity to a star that is allowed to feed that system energy. Because without energy, you go nowhere. The problem is, well, the good thing is that the energy from the sun is free. The problem is it's not accessible, right? And so we need a way to be able to tap into this free energy. And the way we do it is through a process called photosynthesis. So plants are the only ones, and algae, some algae do it, some bacteria do it. who can do photosynthesis. And basically, this is a process that's job is to harvest energy from the sun, and it converts to chemical energy. So it takes the free energy from the light, which is a type of energy, and it converts that energy into a chemical energy, which is something that we can use. Okay. And so that's kind of what it does. And so what it ends up producing is things like sugars, glucose in particular, which is an energetic molecule, has chemical bonds in it that are full of energy. And this is kind of the beginnings. This is the foundation of our food chains. So when the cow is eating the grass, the cow is eating the glucose, the sugars that have all that energy packed into it that the plant captured from the sunlight. Then the cow's metabolism will convert that energy from the plants into and use it for its own body and infuse its own tissues with energy. And then when we go and eat the cow, then we take the energy that's in the cow and then we pack it into us. Make sense? Okay, so that's that one. We'll have more to say about that later on. Now, the other one is homeostasis. Now, this is the one that's kind of confusing, right? But basically, simply put, this is where you're essentially creating a constant internal environment, okay? And so the reason why you want to maintain a constant internal environment is because it basically allows sensibility to your internal mechanisms, right? So for instance, it allows your metabolism to be able to have a reliable environment. Think about it. If you had different types of things happening in the body that were constantly peaking and crashing wildly all over the place, it'd be difficult to do something with that, right? It'd be difficult to function because you would never know what you're waking up to any given day. I liken it to like living with a person who's very volatile, emotional, right? So like they'll blow up and just like go totally Quasimodo on you, right? Or they'll just be sweet as all get out. And the problem is it's like either really, really high or like really, really yikes. And it's like on any given day, you have no clue what you're dealing with. It would be difficult to be functional if that's the reality you had to work with, right? So what homeostasis does is it smooths those spikes and troughs out so that on any given day, and that's kind of what, for people who have those really, mental health issues like that, a lot of times I take medications that kind of takes the highs and makes them lower and takes the lows and not makes them so deep so that they're kind of like a little more even keel. And what that means is every day you wake up to something that's a little more predictable. So now it's like, okay, now I can function because I know roughly what I'm going to get. Okay. And that's kind of what homeostasis is. Your body basically wakes up and says, okay, I can function now because I have a relative expectation of what my environment's going to be like. That's the importance of homeostasis. So response to stimulus. So response to stimulus is important, right? Because ultimately you want to be able to adapt to your environment as you pick up those stimuli. So for instance, if you were to take a look at an external stimulus, a good example of this is putting your hand on a hot stove. The first thing you need to be able to do is you need to be able to have a sensory strategy to be able to sense that this surface is hot in the first place, right? Then what you want to do is you want to relay that information to like an integrator, right? Or an interpreter. So in our particular case, our interpreter is our brain, right? So our brain is the one that receives the stimulus and says like, oh, okay, I'm interpreting this as quite hot. I probably need to do something about this. And then the interpreter will send some sort of a message to an effector that will basically do the job. So for instance, you have sensors that tell you that the hand is too hot. Your brain is actually the one that determines that, right? That puts the value judgment on that. It's like, I feel the heat is about here, it is what it is, but this is too much for me. So I'm going to send a message now, a signal to my hand, to my effector, my muscles in my arm, to move. Otherwise, I'm going to burn, right? And so you have to have that kind of relay system. You have to be able to respond to stimuli, to be able to interpret what you're picking up, and then to be able to act accordingly so that you can adapt your environment. That's critical. So external stimuli is stuff that's happening to us from outside. You also have internal stimuli. This is basically adjustments that you're making right now that you're not even aware of, right? For instance, you're making constant adjustments to things like blood pressure because throughout the day, your blood pressure is constantly peaking, troughing, peaking, troughing, peaking, troughing. And when your blood pressure gets high, you've got physiological mechanisms that turn on to deflect that curve back down. When it gets too low, you've got another mechanism that inflects it, sends it back up, and so forth. It's kind of like physiological table tennis. That's like you get too low, you send it the other way, get too high, you knock it back the other way. You're kind of got like pong, right? And so that's kind of what homeostasis works like. No, the thing is, you have to have sensors to sense your blood pressures as it increases or decreases. Once you have that sensation, like, ooh, the blood pressure is getting a little too high, we need to send some sort of a signal to the brain to do something about that. That's your interpreter. And then the brain has to send some sort of response out to your body to say, you know what, you need to do this. Turn on your mechanisms to turn that curve back down. Okay. Pretty much anything that you can measure in your body is under homeostatic control. So it is a main thing in your body. Now then, when we take a look at reproduction and development, obviously, of course, reproduction is adding to your next generation. That is the holy grail. But in order to get there, you have to actually create a reproductively competent body, which basically means that going from 0, which is one cell with 46 chromosomes, ultimately to a little baby. It's like a little diaper I put on there. By the way, we don't come out with diapers. Those diapers not included. You got to get those on your own, just saying, in case that was a mystery or not, or you had questions. There it is. Right, so we have to grow and develop. So what are you developing from 0 to baby? You're developing your organ systems. Those are all developing. And then when you're born, you don't stop, do you? Because you're not ready to reproduce yet, are you? Does a baby reproduce as soon as it's born? No. You don't reproduce for another decade-ish or so afterwards, right? Technically speaking. Biologically, the body is capable of reproduction at what age? Roughly about what age? Yeah, puberty-ish, right? And there's a little bit of a range in there, right? But basically puberty. Because basically as soon as puberty hits in the female's body, the ovaries start producing estrogen. She starts producing follicle-stimulating hormone, luteinizing hormone, which are all part of two things that are happening in her body. Number one is her gametes are getting prepared to ovulate. So those are developing those eggs that she was born with. They're all born with the number of eggs they'll have throughout their entire life. And so those hormones will start to develop those eggs, and then they'll ovulate them every month. And then you'll just basically do that next cycle. So that's the next egg up, next month around, the next egg up, the next month around, and you do that until you hit menopause. Okay. The estrogen is feminizing the female body, right? Kind of sculpting the female body to create those secondary sex characteristics, not just the breast and the pubic hair and things like that, but also the shape of the body, right? I mean, if you take a look at the shape of a woman versus the shape of a man, the shape of the body is different, right? A woman has much more of a kind of a sleek sort of I don't say curved, like, I mean, it's a kind of more of an agile sort of a shapely look to it, right? Whereas male body is more bulky and thick. The musculature is kind of bulky, right? And kind of broad and big and brutish looking, right? So that is all driven by our hormones. Estrogen for the females. In the males, the testicles start sending out testosterone in massive amounts. And so the testosterone is what masculinizes the male body. That's what turns little boys into men. And it also is the hormone that's responsible for sperm production as well. So at puberty, we're capable, biologically speaking, of having babies. But probably most people would not advise a 12-year-old to have a baby. Because for us, there's more to just babies than having them, right? So we're not the kind of species that just drops a baby and goes, right? That's just the beginning. We have to take care of the baby, defend the baby from predators, and a 12-year-old can't do that very well. When you're 18, you can do it much better because you've learned a lot more and you've had more experience. So you're a little more savvy. In your 20s, you've learned even more, right? And so that's kind of how that works. So we basically will develop, we stop growing, by the way, around late teens or so. But we never stop developing. We're developing in different stages in different ways throughout our entire life. We go through different stages. We have teenage development. We have early adult development. We drop into a different developmental stage when we become middle-aged. As we get older, into older adults, we drop into yet another development stage. So we are developing from beginning to end in different ways and different stages. So that's all reproduction. And then, of course, part of that reproduction is our DNA. We'll talk a little bit more about this. So when we actually pass on our traits to other generations, we're passing on our genes, which control our traits, to other generations. And with our genes, which basically are just short segments of DNA. So this is basically, DNA is all the information that we'll ever need for living organisms. I always like to think of DNA, like what's inside your nucleus, as everything necessary to build you from scratch. That's a lot of information, a lot of details in there. And that's what you pass on to the next generation. So each gene is controlling a particular trait. For instance, for your hair color, you'll have a particular sequence of DNA that basically determines your hair color. Another gene or another sequence of DNA will determine your eye color, and so forth. Okay, and you pass these on. Now, what happens if you change that sequence? Well, if you change the sequence, then what you've created is what's called a mutation. So this is a variation in genes, and generally speaking, genes are mostly negative. It's very rare. As a matter of fact, I don't think in all human history we've actually documented a true bona fide beneficial mutation. But we have tons of negative ones. We have big old thick books. Actually, for my genetics class, I got a big old book of human genetic disorders that thick. It's like 8-point font, both sides. I mean, it's like tons and tons of genetic mutations in there. All bad. Nothing good happened there. So we have tons of bad mutations. Not a whole lot of evidence of good mutations. So mutations by an overwhelming majority are negative. But the problem is, and this is kind of one of those soft spots of evolution that I always have no hesitation of poking at, because even though I got my degree in evolution ecology, I always like to sort of poke at the softer spots of it, because a lot of my colleagues ignore those, and that's not a good thing to do in science. You want to keep working on the things that you need to work on, not just ignore them. But mutations is one of them. Why? Because mutations is one of the big mechanisms of evolution. And the idea is that all of our traits basically came by way of mutations. But the problem is they're so rare to get a beneficial one that we would have to have been around for a very, very long time in order to accumulate the number of mutations we would need to be able to make a functional organism. Okay. And so there's a lot of problems with mutations. Not only that, but mutation rates are notoriously unreliable. They change from species to species, from actually cell to cell, from gene to gene. So it's very difficult to sort of pin down a mechanism using mutation of evolution, because mutation rates are just notoriously just like difficult to deal with. And that's before you get to the idea that beneficial ones come along so rarely that it would require probably billions of years for this many mutations to come. Mean that they didn't. It just means we don't know how this worked. There's a lot of things that we need to consider. It's like maybe mutation rates weren't always the same in the past. That's something we need to consider. And so there's a lot of different ideas that we use in association with this. But originally in evolution, mutations were like the mechanism for evolution, but it's a weaker mechanism for evolution, I think, now that we've learned a lot more about how things work genetically and also how things work evolutionarily. Good. Yeah, actually what you're talking about is the bullseye. I want to go through this quickly, because even though I love getting bogged down in evolution, I don't want to get bogged down in evolution for us, because we're human biology, not evolutionary biology. But there's two fields of evolution, generally speaking, that we look at. One's called the gradualist view of it, which is essentially all life descended from a common ancestor through incremental change, one beneficial mutation accumulating after another, and that every split off the tree was accumulated because of a beneficial mutation that created a new species, and then you kind of just keep adding those on over generations until you get the species that you see now. That's one. And then there's another version of that came up in the 70s called punctuated equilibrium. And so a punctuated equilibrium was an answer to a problem with the gradulus, because the problem with the gradulus is that when you have this sort of gradual sort of transition from one species to the next, then in the fossil record, which is our main evidence for these species that are no longer around, we would expect to see a high percentage of what's called transitional fossils. That is to say, the transitional stage from species A to species B, because it would be a slow, gradual journey through that transition. We would expect to see a lot of those in the fossil record. The problem is we don't. We see fully formed organisms, but we don't see the transition between them. And so that was a problem for gradualism, because that violates their prediction. They would predict that there would be the opposite, and that's not what we see. In punctuated equilibrium, which is kind of what you're talking about, their idea is that evolution in many cases isn't gradually changing. What happens is there's no change for long periods of time, called equilibrium points, right? There's areas of equilibrium, and they're punctuated by catastrophic events, species mass extinction events, like what you're talking about, where just the environment goes on meltdown, and mutation rates, instead of being slow creep, like dramatically skyrocket. They get very, very fast. So it's almost like you're self-mutating because you're trying to like play the odds. You're trying to play a number odds. And so what happens is speciation moves very quickly in a very short period of time. And then it goes into another period of stasis where you don't see any change anymore. The problem is the change that happens, what you're talking about, those punctuated speciation events happen so quickly that there's no time for them to deposit in the fossil record. And that's the reason why we don't see them. So what you were talking about is actually what punctuated equilibrium proposes. And so they kind of have two different conclusions to them. That makes sense? But you can see there's a lot of evolution is not like the foregone conclusion that a lot of educational sources will make it seem. Right? There's a lot of soft spots in it. There's a lot of confusion points in it. There's a lot of questions in there that we're working on. It's an active area that we're really trying to figure out. And there's a lot of things we know pretty well. There's some things that we're kind of like, we kind of know it, but it could be other things. And there's some things we're like, wow, this is a guess. I mean, we're just guessing at this one if we're honest with ourselves, right? We don't really know for sure. Okay. And so I like to be honest about those, because I don't think we do just general public a service by not being honest about those. It doesn't make for a nice, pretty story, but it makes for a more authentic story, which honestly I think is more interesting, because there's a lot more going on in evolution than most people are led on to believe. Okay, let's take a look at, of course, evolution. That was pretty much this entire slide. Thank you. So gradualism basically is looking at, whoa, that was a huge skip. Okay, sorry about that. There. So one of the other mechanisms of evolution is natural selection, which is the one that Darwin proposed. Basically, environmental pressures will weed out those who are not adapted to that environment. That's natural selection. And then in addition to mutation and natural selection, we have adaptation. Right? So adaptation oftentimes is essentially the change in a population over time, but adaptation suggests that this is just an adaptation from endogenous genetic variation. That means genetic variation that's already in you. A lot of evolutionary studies, they begin at the family level. And the family level, just to let you know, is if you've ever seen an organism and you're like, that's kind of dog-like, or that's kind of cat-like. I don't know what it is, but that's kind of bear-like. What you're thinking is at the family level, okay? And at the family level, from there on, there's typically already a big pot of genetic variation already in there. And so adaptive radiation probably explains a good chunk of what that is. So a good example is the dog-like creatures. So probably back in the day, there was a dog-like creature at the family level that had a ton of genetic variation in it. And it basically evolved or adapted by environment into Canis familiaris, Canis lupus, that's house dog, wolf, coyotes, jackals, dingoes, based on the environments that they went into. Certain traits were favored in one environment, certain traits were favored in another environment, but they were all kind of weeded out from that original genetic pot that you have at the family level. That's adaptive radiation. And so when we look at organisms today, that probably is something behind a lot of those. Okay. How do we know that? Because we know there's a lot of genetic variation. Because take a look at just one type of dog, the house dog. How many breeds of dog are there? A lot. And everything from a mastiff, which you can ride to work, to a teacup poodle, which you can have in your hand, and everything in between. And all those are one species. Not even getting to the family level. That's just what was in one species. That's how much genetic variation was in one species. So that's a lot of genetic variation. So that's kind of like one of those things where a lot of times people will talk a lot about mutation and stuff like that, but I'm like, you don't need, from family on, you don't need a lot of mutation or a lot of extra information. Because if there's that much information in just the basic house dog, just the pet, to create such vastly different types of dogs, then you can only imagine how much variation is in the family level. I mean, just unimaginable amounts of variation. You can literally just spit off any kind of a dog lineage that you want, right? Without even having to do another mutation from then on out, right? So there's a lot of that kind of goes in there. And this is kind of what it looks like. So here's kind of how we set up these relationships. So typically what will happen is we basically take organisms and we cluster them together by similarity. The idea being, and it's kind of like the way we do SIB-ships, right? So we basically say that, and this is kind of the way evolution works. And this is the way it worked most in the 19th century. It's a little more better. It's better now than it was back then. But we still have a lot of holdover from the 19th century, which I think still needs to be updated. But this is how it works. The way we organize these, what's called phylogenies, these relationships, is we take things that look similar. The idea being the more similar you look, the more likely you're closely related, which makes a lot of sense, right? If you take basically some two people that are from the same ethnic region, then they will have some resemblances to them, right? So for instance, you can take a look at somebody and be like, oh, you kind of look German, or whatever that means. But that's because there's a lot of people in Germany. They come from a similar population, a similar culture. They share a very similar a genetic background, that Germanic background. So culturally, population-wise, they have a lot of similarities there. And so you can be like, okay, so you look kind of German, as opposed to Spanish or Portuguese, right, or something like that. So you can kind of tell a little bit just by the way they look that you're kind of close. That doesn't necessarily mean that they're that closely related, but as they get closer related, they look more and more alike, right? So for instance, I look very close to my cousin, more so than other people who are of German descent. I look even more closely resembled my brother because we are from the same family. So you can see that as the relationships get closer and closer, our resemblances, our similarities get stronger and stronger. That's the same idea that goes into constructing evolutionary phylogenies. So what we do is we basically collect all these organisms and we organize them by similarity, with the assumption that the more similar you are, the more closely related you are. Okay, and that's kind of the basic idea. The important thing to remember is when we're building those evolutionary trees, the one thing that oftentimes, and I think they should be honest about this, that evolutionary biologists never tell you, is that what we collect is this stuff right over here in the red bubble. All this stuff right here, all this doesn't exist. Because these are things that we are postulating were existing back in time. In some cases, we may have a fossil for this. In many cases, we don't. They're theoretical ancestors that we don't have any evidence whatsoever actually existed on this planet. And that's an important idea to remember whenever you're seeing a tree. Doesn't mean it's bad. It just means there's limitations to where we are and how far we can think about these things because we still have a lot of work to do. What that does then, it puts it on us to go out there and say like, okay, we need to find a fossil that connects these trees together. We need to find the evidence. This is our theory, right? This is our idea, our hypothesis. And so if this fossil is the common ancestor of these, then we should be able to see this in the fossil record. That's kind of our experiment. And so the idea is at some point, if you don't find this fossil in the fossil record, at some point you either keep looking or you have to modify your hypothesis because maybe the ancestry is all different. by the way, just because I look like other people of German descent doesn't necessarily mean I'm related to them. Just because you look like somebody else doesn't necessarily mean you're related to them. How many of you guys love to go on Google or something like that, and you love to look up those doppelganger things, right? People who look like other people. Robin Williams and Bono, for instance, right? I'm sure Bono loves that one, but right. So they look a lot alike, right? Do you guys kind of go just sort of brain dead dive on those things? They kind of pop up on your phone all the time. Be honest, I know better. You guys totally go into that stuff, right? Yeah, because it's fun, right? But that's the thing. That's like, just because Bono and Robin Williams look similar, it doesn't mean they're anything closely related. As a matter of fact, they're not. I mean, Bono is Irish and Robin Williams is not, right? I mean, they're completely different. So just because you look alike doesn't necessarily mean that you're related. It just means you look alike, right? So that's a caveat to this whole kind of the way we sort of set these things up. And that's kind of where I kind of push back on that 19th century view of things, where it's like you put all these things together and you set up the relationships that way. Because now we're doing it a little bit better. Because if you do a genetic study on Bono and Robin Williams, you'll see right away, yeah, these guys are nothing similar. They don't even have the same gene background, genetic background, right? So you see it very, very clearly. So when we take a look at these relationships, what we basically do is we set up what's called taxonomy. It's a field of science, and this is basically the classification of organisms. By morphological similarity. So typically speaking, we'll sort of divide these guys up into groups. It's kind of like 20 questions, right? So when we divide these up into groups, they kind of form essentially almost like your species signature, if you will. And so we start off with very, very generic things. For instance, at the very top, organisms that are very similar. So we typically will start off, first of all, with the concept of the domain. In the domain, it's kind of like 20 questions. If everybody's ever played 20 questions, right? You have 20 questions to be able to figure out who I'm thinking about. And so the way you ask those questions is you ask them in binary format. Like, it's either A or B. Like, are you this or are you that, right? So you can kind of whittle it down. The first question is, are you a prokaryote or a eukaryote? Now, a prokaryote and eukaryote are two different types of cells in the world. Every living organism is only going to fall into one of these two categories. Now, typically speaking, a prokaryote is typified by the fact that they do not have a nucleus. Eukaryotes do. That's the big one, okay? So in this, you'll define three different domains. One domain, which is prokaryotic, is archaea. And so the other one, which is also prokaryotic, is kingdom bacteria. So all those bacteria that we kind of wash off our skin and things like that. The third one, which is domain eukarya, basically houses all of those organisms and cells that have nuclei in them. They have a big membrane-bound nucleus in them that holds DNA. And there's major types of, the next level is kingdom. So the next level that you ask, question you ask at the kingdom level is, how do you get nutrients? So you have a different pattern for each one. So kingdom protista is typically full of single-celled organisms, typically, that will do several different types. So they will absorb nutrients, they will eat nutrients, they'll make their own. A lot of these guys are either animal-like, they consume their nutrients, or they're plant-like, they make their own nutrients. And so there's a whole spread of these guys. And so they're kind of like a miscellany of all different types. They can form colonies, for instance, kelp forests. How many of you guys have ever seen a kelp forest in the ocean? Kelp forests are actually colonies of algae or algal protists. Okay, so they're plant-like in nature because they make their own food. But it's kind of a miscellaneous group of protists. Giardia. Anybody ever dance with Giardia? Anybody ever attempt to not dance with Giardia? Right, so you're boiling your water up in the mountains, you're treating it. That's because Giardia is a protist. Happens to be a parasitic protist, but it's a protist. So the other one is kingdom Plantae. So in this particular case, these guys are what's called autotrophs. They make their own food. Photosynthesis. And so that basically creates kingdom Plantae. The mushrooms are in kingdom Fungi. So are the molds, so are yeast and things like that. So these guys will actually absorb their food from the environment. Totally different way of doing it. Okay. And then of course there's us, kingdom Animalia. In this case, we are what's called heterotrophs. We have to ingest our food. We can't go out there and make our own food. We have to actually go and find it. We have to eat it and consume it. And so that sets apart the different kingdoms. And so within Animalia, we're going to focus on Animalia. You can further subdivide us, and we do in the different taxonomic divisions. And so you can divide us into whether or not we have a backbone. That's another big question that we see in taxonomy, right? So are you a vertebrate or an invertebrate? So an invertebrate basically does not have an internal skeleton. A vertebrate does. So for instance, earthworms, insects, mollusks, right? So shellfish, right? So like crabs, things like that. Those are all invertebrates. They don't have a backbone. Anybody that doesn't have a backbone is a vertebrate. If you're a fish, a reptile, amphibians, birds, turtles have backbones underneath their little shells. Those are all vertebrates. We have a vertebral column, right? Like your spinal column. Why do you have a vertebral column? Because typically it's protecting something very valuable. In this particular case, it's protecting our spinal cord. So that is a very, very sensitive part of our nervous system that we have to protect. If you didn't have protection of your spinal cord, life would be very painful. And so that's not a good thing if you're an organism and you're having to live with that kind of pain. Anybody who's had direct contact with the spinal cord knows exactly what I'm talking about, right? It is incredibly painful. And so you can further subdivide the vertebrates, us, into mammals or non-mammals. So what is a mammal? A mammal typically will have hair and fur, but not all of them. But all of them will have mammary glands. That is basically you feed your young with mammary glands. Okay, that's why we're called mammals. And so, for instance, humans, raccoons, seals, all the cute furries with little ears and muzzles, right? Those are all mammals. And so you can kind of keep further subdividing them over and over and over until you get down to a specific species where you have Homo sapiens, okay? Or Canis familiaris, that's just your common dog pet, okay? And so you have all these different subdivisions throughout taxonomy. So let's take a look at the scientific process now. Basically in science, we need to understand, essentially science is focused on how to look at and study the natural world. So it focuses on and only has touch on natural law. So that's all it can do. Can't do anything else, right? The important thing about science and the critical thing about science is that in science, you have to maintain your objectivity. That means science only dwells on data and information, right? Direct inferences and conclusions from discrete data. The one thing you cannot have is subjective judgment. So basically, you do the best you can to eliminate your bias. Not opinion, not judgment, not subjectivity, but objectivity based on data. 2 + 2 equals 4, right? That's discrete and objective, not up to subjective judgment, right? So it'll always be 4. It is what it is what science does. It doesn't interpret things. And in science, unlike the other major intellectual disciplines like philosophy and theology and things like that, science has its own method in order to help maintain this intellectual rigor and to maintain the logical progression of how to actually dissect natural phenomenon, called the scientific method. The scientific method begins, first of all, with observation. So in observation, basically you observe the natural world and you record what you see. You typically are using your senses like sight, smell, and things like that. You could use instruments, tools like a microscope, for instance, or through research. But ultimately, you're making some sort of an observation. Imagine that we go for a nature walk, and we're going for a nature walk with sort of a medieval type of a guy, pre-science, right? So pre-science as we know it. and he's going to make an observation. So his observation is he sees a plant and he notices that the leaf is green. That's his observation. Okay. The problem is he's on this walk with a poet who sees the same thing. The leaf is green. So they see the same observation. Now, the important thing is not what they see, but what they do with it. The next step is going to be a question. Before we get to hypothesis, the next step in scientific method is you ask a question based on your hypothesis. The poet and the pre-scientist is going to be asking a different question. The poet asks, why is it green? But he doesn't really care why is it green. He's thinking big, right? He's kind of thinking like, oh, the ode to the green, right? It's like, all the gods have made it green because green is the greatest color in the world or whatever, right? So that's not the same thing as the pre-scientist. The pre-scientist wants to ask a different question. He's like, why is it green? To say, why isn't it some other color? Why not blue? Why not red? He's asking a different question. What he's after is like, what is the mechanism that makes it green? How does it work? How does greenness work to create green? Not the gods, not all that stuff, right? That's theology. But he's thinking, what is it exactly, mechanistically, that makes this green? That's what he's asking. It's a different question. Now, then the hypothesis is basically going to be your best explanation. It's essentially your educated guess to answer your question. Now, remember, this guy is pre-science, so we don't know anything about chloroplast and chlorophyll and photosynthesis or anything like that. So he doesn't know a lot, but he has a classical training, so he knows a lot about literature and especially a lot about art, because that's kind of what you learned about back in those days. So here's his hypothesis. Ready? Maybe it's painted. That's what he knows. Remember, the hypothesis does not have to be accurate. It does not have to be correct. It just has to be testable, right? That's how we learn. This is the best thing we've got. This is all the knowledge we have. I know about paint. I don't know anything about chlorophyll, right? So this is what I've got. Can I test it? Sure I can, right? That's all I need. So then what happens is then you set up testable predictions, which is the next step. It has to be testable. This, by the way, is a hallmark of science that no other intellectual discipline has. Philosophy does not require you to have testable ideas. Theology does not require you to have testable ideas. Only science requires you to have testable ideas, which is the reason why science cannot say anything whatsoever about anything in the theological ring. Because the concept of theology and deity and the gods is handled by nature, natural law. It is supernatural law, which means it's fundamentally untestable. Therefore, science has nothing to offer or say about anybody's beliefs about deity or theology or philosophy. It fundamentally cannot. It's not designed that way. Generally speaking, in science, we'll use a couple of major types of logic. One is called inductive reasoning. And inductive reasoning is essentially where you have lots of observations that lead to a conclusion. Here's a good example of inductive reasoning. If I were to ask you, what color is the sky without looking, what are you going to tell me? What color? Blue. Are you guys pretty confident about that? Yeah. Why is it? Did you do an experiment to test its blueness? Why are you so confident that it's blue? Yeah, you see it every day. Because by this time in life, how many times do you think you have seen the blue sky? Millions of times? Uncountable, right? Probably more than millions of times. Has there ever been one of those observations when the sky wasn't blue? Nighttime doesn't count. Forest fire territory doesn't count. I'm talking the actual color of the sky, right? Sunsets don't count. The actual color of the sky. Has there ever been an observation where it's not blue? Not cloud cover. It's always blue. How do I know that? Because you guys are really confident about that. You just spit out blue. So that's inductive reasoning. Through the number of observations that you've seen, you make a conclusion with confidence that the sky is blue. Not because you did an experiment on it necessarily, but because you've never seen the alternative to be true. That's inductive reasoning. Now that you've got your testable predictions, the next thing to do is to set up your experiments to test your predictions. So the testable predictions for our person, he's thinking that maybe it's painted. These typically are if-then statements. So for instance, if it's paint, then it should rub off, right? If it's not paint, then it won't rub off. Those are your predictions. If-then statements. If this is true, then this should happen. I should expect this. Then what you do is you set up your experiments to actually do the test. In this case, it's rubbing off or trying to rub off the paint. Okay. Now, whenever you're doing an experiment where you're generating data and from the data you draw your conclusions, that is what's referred to as deductive reasoning. So a lot of science is deductive or inductive, mostly deductive, where you generate data and from that data you draw conclusions. That's deductive reasoning. So when we're setting up our experiment, let's take a look at it. So typically speaking, you have a lot of different variables in your system. And so in able to really pin down which variable you're studying is the culprit behind your change, you have to set up two different groups, like for instance, a test group and a control group. And these two groups are exactly the same, with the exception of your experimental variable. So that's the only one so here's a good example. Let's imagine that I'm selling you fertilizer. And what I want to do is I want to run an experiment to show you that my fertilizer is actually allowing your crops to grow. So what I do is I take a plot of land, I split it in half, and everything here is the same, right? Soil's the same, sun is the same. So the sun is the same here. It's the same rainfall, same everything. And so on one side, I basically get a lot of good growth. All I've changed here is I added fertilizer. On the other side, not so much. This is the side that doesn't have fertilizer. So this would basically be the experimental group, and this would be the control group. Notice everything is the same between the two, with the exception of the variable I'm looking for, which in this case is the fertilizer. The reason why I do that is because if I didn't do that, and I showed you the results, I said, okay, here, look, these crops really grew tall. You come back at me and say, how do you know that wasn't the potassium in the soil? Maybe you had more potassium on one side than on the other. How do you know that wasn't the rainfall? Maybe one side got more water than the other. How do you know that wasn't sunlight? Maybe one side was in the shadow than the other. Any of those could have been the reason why your crops grew. It had nothing to do with your fertilizer. So to do a controlled experiment means you eliminate all those variables. You keep them all the same so that the only thing that changes is the one variable you're studying. So that if you see any data, the pattern of the data is only attributable to the experimental variable that you're looking at. Okay, that's a controlled experiment. We can also use not just controlled experiments, we can also use what's called model organisms, things like mice, for instance. We use that a lot in research. This allows us to be able to do experiments on a similar organism to us, and we can take that information and transfer that to humans. This is how we do drug development, by the way. Most of our drug development has been done using mice, because mice are mammals that are very similar to us. We can create human models of mice of the same disease that we suffer from. The idea being is if I can administer a drug to this mouse and it can cure the mouse, there's a really good chance that if we move this into humans, we could cure humans with it. And that's the way we use model organisms. We use them to understand things so that we can directly transfer that to us. So we'll collect and analyze data, and we'll present our data into graphs. So here's a good example of a graph. So you have your X and Y axis. In this particular case, we looked at blood cholesterol levels as they went up and down over the different weeks. We can also use statistics, which is what these little bars are, to tell you how significant these numbers are. And so that's one thing we also do with data. So if we have a lot of data, we have to basically prove that our data is actually valuable, right? And so we use statistics to interpret our data. We usually will add some sort of what's called a standard error. So like, what is the error here? If it's a big standard error, then that means your data is going to be less powerful. If you have no error at all, that means your data is telling you straight goods, right? And so we kind of describe our data, and we like to also talk about the significance of our data. Like, is it actually just statistical scatter, or is this actually meaningful, right? That's statistical significance. We usually use probability tests to do that. Here's a good example. We're in the middle of an election year. And so we see this in polling all the time, right? So you'll see different types of presidential polls, so-and-so is running a few points ahead of so-and-so. And then down the bottom, you'll say the statistical scatter is plus or minus 2 or 3%, right? So that means if these two individuals are running within 2 or 3%, that's the error. Then technically speaking, statistically, they're pretty much neck and neck, okay? So if it's significant, then somebody would actually be far ahead, well beyond the error. And that's what the probability does. So what's the probability that this is significant? That's what probability tells you. Then, of course, if we have a good idea, we publish it, and then everybody gets to learn from it. Then we basically develop our conclusion. We get to say something about our hypothesis, and we get to sort of describe what our answer is, and we publish that so that the general scientific community can learn from that. So that's kind of how we develop scientific theories, right? Every theory starts off as a hypothesis. A hypothesis that's been well experimented on is effectively becoming a theory. A theory is essentially a hypothesis that's been supported by experimental evidence. And there's a whole range of scientific theories. There's some that are a little weaker that we're still doing a lot of work on. We still have to generate a lot more data in order to kind of get those to solidify. We don't really feel too confident in those. And then there's other theories that we're pretty good about those, right? So we feel pretty good about those. Things like the atomic theory, for instance, the idea of atoms and things like that. So we feel pretty confident about those, even though I mean, a lot of times people say it's like, oh, it's just a theory, but theories are basically hypotheses that have a lot of evidence to varying degrees. So we have theories that we're still actively working on, theories that we're pretty confident about because they've got a lot of experimentation on them. Cell theory, for instance, is another theory that we're pretty confident about. This one is pretty good. So there's not a lot of pushback on this one. But every theory, depending on the theory type, has its soft spots. It has its issues and points where you can kind of push back on it. Some areas where we need to sort of work on a little bit more and kind of tighten it up a bit more. Then we have the concept of laws or principle. Now, typically in biology, I would argue that laws don't exist for the most part. Why? Because in biology, we are designed to be flexible and adaptive, not constant and unchanging. A law, I would argue, is constant and unchanging. A good example is the laws of physics. Those are basically the fabric, the universal fabric that holds our universe together. For instance, if I were to drop this iPad Pro or let go of it, what's going to happen to it? It's going to go down, right? That's the law of gravity. That gravitational law is necessary in the universe to be able to hold our universal reality together. It's what holds the planets together and keeps them from flying out into space, into debris. And it's basically what creates the entire framework of how our entire solar system works because of the gravity of the sun capturing and holding our planets in orbit. So the law of gravity is a fundamental truth that has to exist in order for our universe to even exist. Without it, we have no universe. It literally comes apart at the seams. That's a law. Evolution is not a law. It aspires to be one, but it will never be one because there's just too many caveats. There's too many soft spots in evolution that we need to work on. And certainly is not universal in the sense of a law. That is to say, it doesn't hold the universe together. Why? Because evolution is a biological theory that's designed to be flexible and adaptive, not constant and unchanging. Laws tend to be constant and unchanging. And evolution is certainly not constant and unchanging. How do you know that? Because if it were, there wouldn't be two versions of evolution, gradualism and equilibrium. Right? So you wouldn't have multiple different types of pivot points with evolution if it were a law. So for that one, I disagree with Mater. Okay. So when you take a look then at your controlled study, you have a hypothesis, an example of one where we have an antibiotic. So we're going to use antibiotic B versus A, and we would test to see whether or not A is better than B or B is better than A. So what we're going to do is we're going to basically use a control group. That is essentially an individual who gets a placebo, which is essentially a pill that does not have any medication in it. So it's kind of a neutral pill. And then you're going to have two different experimental groups. Those two different experimental groups, one is going to have antibiotic A. The other one is going to have antibiotic B. So you have three groups. Now, we oftentimes do this in clinical studies for drug testing, drug development. That is not drug testing, you're testing for drugs in your system, right? But for drug development, is we do what's called a double-blind study. That is to say that nobody knows what they're getting, not even the doctors. So the doctors don't even know what they're prescribing. The patients don't know what they're prescribing. That's the two levels of blindness. The first level is the patients don't know what they're getting. The doctors don't know what they're getting. So it's double blind. The only people who know what they're getting are the people who are processing the data. And that's it. So that allows basically the removal of bias. And this is particularly important in drug development. You know why? Because what happens as soon as you take something? Oh, we are superstitious creatures, right? You know how you know that? The supplemental industry is full of this stuff. Oh, I took this, therefore it worked wonders. Actually didn't do anything because you just kind of like psyched yourself into it. You think it worked and therefore you believe it worked, but it didn't actually do anything for you. You just felt better about yourself because you thought it worked, right? So that's an uncontrolled, illegitimate study. This is designed to remove that kind of bias that we have. So then what you do after that is you basically process this. So essentially you give each group their pill. So here you can see your placebo group, your group A, your group B. And then when you process the results, you can see that antibiotic B did better than antibiotic A. And you can see you got a little statistical error measure right there. So it's significant. And so ultimately then that's kind of how we do medical as well. So let's just take a look at some, just a couple of ideas just to sort of wrap up the day in terms of science and society. So science basically is our sort of method-driven way of understanding the natural world. Technology is the application of that science, right? So for instance, we learn a little bit of physics and we apply that to our technology to create iPhones. And so we're really good at that, adapting our knowledge to technology. Now, one of the things also that we have that's facing society is obviously climate change. So clearly we have some climate change that's happening. So for instance, we have CO2 buildup into the atmosphere, and CO2 is what's called a greenhouse gas. That is to say that it traps heat and re-radiates it back down to the earth, abnormally heating up the atmospheric temperatures, and that kind of creates global warming. That's because of the increase in CO2 levels. And so what that does actually is when you re-radiate that back to the earth, is it starts to warm up the water. And so the problem with warming up the water is the oceans themselves are the major drivers of our atmosphere. So when you take a look at an El Nino year, which creates different rain patterns and things like that, the reason why you have El Nino is because the surface temperatures of the ocean just off of Ecuador, I believe, abnormally increases in temperature, and that just changes the atmospheric conditions. It puts more moisture in the air, changes the weather patterns, And then the entire atmospheric outcomes change for everybody who's kind of in line for those weather patterns, including California as it moves across the country, to us even. And so it changes all those weather patterns in ways that we can't predict. In some cases, some places will have heavier rain seasons, and others it'll be more desert-like. And so it's hard to predict, but we do know that the warming of the oceans in particular is problematic for changing atmospheric temperatures, and it could change them in ways that we don't want, right? Creating desertification in areas that aren't deserts, and also wreaking havoc on agricultural areas that we rely on. Okay, so if you turn our breadbasket central Midwest area, the agriculture area, into a desert, a lot of people would go hungry. Right, so that's the reason why we're watching that, is because we don't want to do anything abnormally bad to our environment. And so we have technology, we have biodiversity, which is essentially the diversity of species, and of course extinction, which is the death of a species. And that's always a bad thing, because we never know what the death of one species is going to do to all the rest of them. Okay, so that if you pull one thread, it could undo the entire tapestry. And that's what we're worried about with extinctions, is because we don't know when one extinction is the beginning of a mass extinction event. And mass extinction events are horrible to go through. We've never been through one, and likely we will not survive one if we do go through one. Most species die in a mass extinction event. Very few make it through. Like this little guy right here, our ring-tailed lemur. You can see these guys at the Denver Zoo. So these guys are in danger. You can see them on Madagascar, but they're very, very endangered. So they're kind of cool to see at the Denver Zoo. Okay, so there's that. That is chapter 1.